Good afternoon and welcome. I'm Chris Charton. I'm a member of the Board of Directors at LJI, the La Jolla Institute for Immunology, and I'm also chair of the Leadership Committee for our Center for Infectious Disease and Vaccine Research. So on behalf of the entire board and all the folks at LJI, we'd like to welcome you to our latest edition of our webinar series, Live from the Lab. Today's program will feature two of LJI's faculty members who are both preeminent infectious disease experts, Dr. Erica Ollmann Saphire and Dr. Sujan Shresta. As a quick overview of today's program, it will include an introduction to our President and Chief Executive Officer, Dr. Mitch Cronenberg. Then we'll have the presentations from Dr. Saphire and Dr. Shresta. They'll be followed by closing remarks from our Executive Vice President and Chief Operating Officer, Steve Wilson. And then we'll be wrapping things up with the Q&A session. So I hope you'll stick around for the entire hour as it promises to be an intriguing discussion. And now I'd like to introduce Dr. Mitch Cronenberg, our President and Chief Scientific Officer here at the La Jolla Institute for Immunology. Not only has Mitch served as President and led LJI for the last 18 years, but he is also a world-renowned scientist in his own right. So Mitch, the virtual podium is yours.
Thank you very much, Chris. We're grateful to all you do and the other board members for helping to guide our organization. Claire, next slide, please. So a few facts about us. I know some of you are very familiar with the organization and some less so. We were founded 32 years ago and have almost 500 employees. Our research, which is our raison d'etre, the reason why we exist, is organized into 21 laboratories, each led by a principal investigator or faculty member who chooses their area of research. We have 172 people who are involved in technical support and administrative support. And we have 213 postdoctoral fellows, individuals with PhDs and MDs from countries all around the world who come to work in our laboratories. Next, please. So our mission is to engage in a world-class biomedical research program with a focus on the immune system. And we want to not only uncover knowledge, but we want to share and partner such that the results of our discoveries will make outstanding contributions to the betterment of human health. Next, please. Immunology is important because it's related to so many diseases and conditions. But in speaking about immunology and in thinking about immunology in our own internal organization, but in speaking about it to the general public, we've organized our principal investigators or faculty into three areas. And they are infectious disease and vaccines, which we'll hear about today, cancer immune therapy, using the immune system to fight cancer, and autoimmune diseases, autoimmunity and inflammation, a variety of diseases such as type 1 diabetes, multiple sclerosis, inflammatory bowel disease, and so on and so on. There are over 80 of these. And each of our faculty is concentrated in one area or the other, and some in their work really fits into a few areas as diagrammed here. Next, please. So when the pandemic started earlier this year, and it seems like a very long time ago, our investigators, and especially those with expertise in viral immunology, organized themselves into a coronavirus task force, working together to gain insights into what is a productive immune response to the virus and what may be a harmful immune response, because a lot of the people who are very sick actually have a harmful immune response. And this group has been extraordinarily effective, including the two speakers we'll hear about today, Erica Ollmann Saphire in the top row and Sujan Shresta in the middle and the bottom row. Next, please. So normally on these webinars, I've talked about some recent findings, and we have many, but you're going to hear from two investigators. I want to tell you about something a little bit different, and that is we have 480 or so employees. How do we operate safely? Of course, not everyone is in the building. In fact, some people are working entirely from home if they can. But we have several hundred people coming to the building every day, and it's so important to operate safely. Our employees, when they enter the building, are asked to sanitize their hands. They have a code, is the step 2 that you can see on the slide, that they have to scan. And that scan indicates when they last received a test for viral genome, a PCR-based test that they're obligated to undergo every 2 weeks. And then further to the left of the step 2 in the big yellow circular sign is a thermal temperature scan for the skin that confirms that the individual doesn't have a temperature. Anyone with any symptoms, of course, must stay home. Next, please. So we've developed a screening test for viral genomes. It's not FDA approved, so we don't use it to make a definitive medical diagnostic. But if anyone
enough doses to deliver to everyone on the world. What's clear is that these vaccines, even though they might get paperwork approval, aren't going to be in all places at all times. There will be people who have not been vaccinated by choice or because they haven't been vaccinated yet. It hasn't gotten there. There will be people who can't be vaccinated. For example, pregnant women, immunocompromised, and infants can't receive some kinds of vaccines. And then there will be those people who were vaccinated, but that vaccine didn't take. And that is often what happens in elderly people or immunocompromised people. And that's unfortunate because these are the people that we most need to protect against COVID-19. The vaccine may be less efficacious in these people, or you might need a higher dose. The other potential worry is, what if the virus changes? With 55 million infections, and spill over and spill back to different non-human species, there could be mutagenic sampling, mutagenic changes, and virus could change. And with enough changes, our own immunity could be compromised. It could be a different virus. So how do we solve that problem? How do we solve the problem of understanding where immunity comes from, understanding how to deliver it, how to cope with the fact that it's going to take quite a bit of time to mobilize vaccines, and even once mobilized, they're not going to be in all people. And of course, a vaccine doesn't work if someone hasn't taken it. Well, a major goal of vaccine is to produce antibody. Antibody, now I'm showing you here a cartoon of SARS-CoV-2, the virus that causes COVID-19. On the surface, those red molecules, those are called the spike protein. That is a molecule the virus uses to attach to your human cell and drive it in. A major goal of vaccine is production of antibody, largely against that spike protein. Antibodies are these Y-shaped structures. They're molecules that your own immune system makes in response to vaccination or infection. They have 2 arms that have hypervariable fingers at the end. And those fingers give us something like fingerprint-like recognition of a particular piece of a particular pathogen. An antibody will hit just one precise site on SARS-CoV-2 or one precise site on measles. And by hitting the right site, it could have the right function. Okay, and those functions are 2. Antibodies do 2 things. They can directly neutralize that virus. They can just inactivate it, like a club in your steering wheel. Viruses can also, antibodies can also recruit immune system help to the site of infection. They can flag that virus for destruction. They can unflag an infected cell to come and clear it so it's no longer a factory to make more viruses. And they can rouse the alarm for greater levels of immune protection. So antibodies, if they hit the right place, can directly neutralize the virus and bring about greater immune protection. Now there's lots of antibodies. A person that has either survived infection or been vaccinated will make millions of antibodies. Among those millions, there are some that are extraordinarily potent, there are many that are okay, and there's some that are not so good. There's definitely needles in that haystack. The important thing we can do, and this is a tremendous complement to the vaccine effort, is by searching among all of those antibodies, we can figure out which one or two or three are the very, very best. You can make those a clinical grade material in a laboratory. You can deliver that as a drug. You can give someone the antibody they wish they had had. You can give them the immune response of a rock star within hours of receiving that through an IV. That's called antibody therapy or immunotherapy. And a really good analogy is anti-venom. Anti-venom is antibody against snake toxin. If you get anti-venom right after a snake bite, those antibodies will find and neutralize that snake toxin, save your leg and save your life. So with antibody against virus, just like antibody against snake toxin, earlier is better. You don't wait 3 weeks after your rattlesnake bite to see how you're going to do on your own. You get the anti-venom right away. It's the same thing. Antibody against virus is most important early in disease when you first found out you're infected and you're still doubling the amount of virus in your body every so often. That's the time when you need antibody to knock it down. Antibody is most effective in early stage disease. But it's remarkable. You can use it immediately to either treat an early stage infection, prevent it from becoming a severe infection, or you can use it for prevention. You can give someone like a frontline medical worker antibody to protect them for weeks to months until they can make that vaccine. Or that's also important protection when the vaccine isn't available yet, or if that vaccine is unlikely to work, like stopping an outbreak in a nursing home. Now here's the challenge. There are a quintillion possible antibodies in human sera. That's a 1 with 18 zeros after it. There are at least 50 different companies racing forward, trying to find the very best antibody that they can find in their own laboratories, deliver that as a therapy. Each one of these companies is using a different strategy to find them, and they've each got different therapeutic candidates. Which ones are best? Which are the ones that we need to put our clinical trial beds and dollars behind? Which are the ones that we need to mobilize into people? And how do we know? Every company has evaluated their antibodies using a different strategy. So the little bit apples to oranges. This week, 2 got emergency use authorization, one from Lilly and one from Regeneron. And that's important. That means the FDA believes that there's maybe sufficient data to say that this could have a protective effect. It's not a full approval, more data is needed, but there's some hope that making this available for very high-risk people could provide some benefit while the rest of the data package is being collected. But of all the competing therapeutics going forward, which ones are best? And can you make an ideal treatment cocktail, a mixture of 2? If you put 2 antibodies together, sometimes they can have a synergistic effect and make it more potent, give it better than the sum of the parts. And so what the Gates Foundation tasked me to do was to work with each of these competing companies and academic labs and evaluate their candidate antibodies side by side in an apples to apples comparison. Figure out which ones were most effective, figure out why they were most effective, figure out which laboratory experiments we could do that would best find them so we can streamline the research pipeline next time. Engineer those molecules to make them more durable, longer lasting, more resistant to mutation, and more potent. If we want to deliver something to millions of people, it's going to have to be potent and easy to produce and make these available to anyone that needs them. And so the workflow is this, where we will ask these competing entities, companies, laboratories, nonprofits, biotechs, to contribute their samples here. We give them all a code name, and that makes the study very fair. We're comparing everything side by side, apples to apples, under blinded conditions with a data-driven decision of which therapies are best and why we think they're best. We will pack identical box sets of the world's therapeutics and send them out to multiple experimental experts here at LJI and everywhere in the world. People that are the best in their field at evaluating some aspect of an antibody function. They're evaluating the set and we're putting the data back together. Then we're building what's at the bottom, the CoVIC DB database. This is an effort launched by Professor Björn Peters here at La Jolla Institute, who's an expert in amassing and analyzing immune data and how immune functions and immune therapies gather. And by having all of the data blinded, we can make it immediately available to the research public. So everybody can learn what we are learning on the data on this set of antibodies as we can. From this, we'll be selecting ideal treatments and cocktails. Now, there's a benefit to the contributors. The blinding and other agreements we've made protect their intellectual property. And we're generating a lot of information in a lot of different ways. That maybe include multiple kinds of experiments that they don't have access to in their own lab. So they can have all of that data back, and they can use that for their own investigational new drug filing. So we want to make these available. If we have better data, they can use it too. So here's what we're going to do. We're going to do multiple parallel experiments to figure out how tightly they bind to spike, where they bind to that red spike on the surface of coronavirus, how they fit into different groupings, which ones are like each other, which ones are different from each other, which ones are vulnerable to mutation, which ones are not. We're evaluating the high-resolution molecular structures of these things, understanding how they recruit immune protection in different ways. We're looking at the ability of these antibodies to neutralize that virus in multiple parallel systems. We're looking at their likelihood by which mutations will escape these, which ones cannot be escaped. We're correlating mutations that we're observing in the world to the binding patterns of these antibodies, so we can make a library of what our medical arsenal is. If a mutation shows up in Wisconsin, we know that these therapies will be appropriate and these will not work. That will help direct medical decisions. And we're looking at the ability of each one of these antibodies to deliver protection in living things, and we're making novel combinations. It might be that the best 2 antibody treatment is a molecule from Washington and a molecule from New York. And we'll never know that unless they're in the same place at the same time. This study provides that opportunity. So what are our goals? Our first goal is to make an effective product. We're going to try to find the most effective antibody cocktail to prevent severe disease, one that's going to be resistant to mutation. But while we're at it, while we have united all of these competing groups and gotten all of the world's immune experts together under this roof here at LJI, can we build a body of information so we don't have to do a pandemic on this scale next time? Can we build a body of information to help us understand what makes one antibody better than another? If we want a 2-component cocktail for synergy, well, what gives it that synergy? What happens when one binds that helps the other one? And what does this analysis of these antibodies tell us about vaccines? We're going to figure out what the very best immune response might be. That's something we would like a vaccine to deliver in people too, and accelerate this pipeline, make long-lasting therapeutics. And so this was the lofty goal that we set out in February. And now the question was, would researchers contribute? They have tremendous investments in their therapeutic molecules. It's tremendously risky to advance them to market. Would they give them to another person to evaluate? And the answer is that they did. And a tremendous effort for advancement of human health and getting us all out of this pandemic, we have had tremendous response. And we now have 209 competing therapeutics that we can evaluate side by side. And they come from academic labs, nonprofits, small biotechs, larger biotechs, and the household name major international corporations on 4 continents: Europe, North America, Asia, and Australia. And so we now have a wealth of samples and a wealth of information. What are we learning? This first bit of analysis I'm going to show you is that we're doing a detailed profile of the ability of every single one of these therapeutic molecules to inspire different kinds of immune protection. And this is work done by my collaborator, Galit Alter's lab at the Raygon Institute. We can also line up every one of the world's therapeutics, and this is now the database created by Bjorn Peter's team at the La Jolla Institute, including Dr. Randy Vita, Mari Kojima, Brendan Ha, and so many others. We can actually rank all the world's therapeutics by how tightly they anchor themselves to that coronavirus spike and how potently they neutralize that virus. This data, as it comes in from our experimental partners, is uploaded and shared with the world immediately. So everyone's learning this as we learn it. We can also look at that landscape of antibody therapies against coronavirus, and we can group them by ones that bind in the same place and have the same function, and ones that bind in different places might have a different function. That helps map susceptibility to escape. If there's a mutant here, these antibodies won't work, but another one would. This is work done by Dr. Dan Bettinger and Tim Garriman at Cartera by doing high-resolution epitope binning for our Coronavirus Consortium. They can map antibodies into groupings or neighborhoods of ones that are like each other, ones that are different from each other. Now, anywhere you see a black line here, that's a competition. That means that this antibody can't bind at the same as that one. And what you see here, there's a lot of black lines. Most of the world's therapeutics all target the one part of that spike we call the receptor binding domain. Now, they have different ways of hitting that receptor binding domain, but that's a little bit of a vulnerability. So one of the projects we've now launched in my lab to complement that effort is to search for antibodies against other parts of spike. We might get better synergy that way, and we might be less vulnerable to mutation if it emerges in that way. But in the meantime, since we now have all of this data on the world's therapeutics and where they bind and how well they work, how do we choose an ideal pairing? How do we put together two in a cocktail? Well, we might look for complementary functions. So for example, if I plot a section of antibodies that we get based on their ability to block attachment of that virus to your human cell, when that red spike protein wants to attach to your human cell and drive itself in, it wants to anchor to a receptor called ACE2. We can rank order the antibodies by how potently they can block that interaction. That's how they neutralize. We can also rank order them by whether or not they do it really well, they're potent, or not so well, not potent. We can find that down in the right corner, those antibodies contribute to the study are the ones that are most potent in neutralization, and they do so by blocking attachment of the virus to its human cell receptor. But if you look in the other corner, there's a handful that are different. They're also incredibly potent against the virus, but they don't block receptor binding. They do something else. So we're now trying to untangle what it is that they do. And this gives us an opportunity to provide a two-sample cocktail. We want an orange one and a green one together to hit the virus at two different footprints. Okay, what does that really look like? One of the specialties of my lab is in high-resolution molecular imaging. So you've seen these cartoons of the coronavirus with the red spikes on the surface. This is what that spike really looks like at the molecular level. This is the high-resolution view of the polypeptide thread, the genetic information that knits together that three-dimensional molecule in space. And this is an image from Dr. Vamsi Rayaprolu in my lab, showing us how that coronavirus spike is knit together and where its component parts are. By looking at how it is assembled as its molecular machine, we can see each of the gears in the part and understand how it works. We can use that information to analyze every sample in our database. So for example, of our 209, let's just look at number 10, antibody CoVIC 10. We can see where it lines up in our database between samples 9 and 11. We can see the type of antibody it is, IgG1. We can see that it maps to epitope class 3A. We can get numbers and how tightly it binds to different versions of spike, whether it binds to the receptor binding domain, N-terminal domain. All those binding constants were done for us by Georgia Tamaras at the Duke Human Vaccine Center. We can look at how potently it can inactivate 50%, 80%, 90% of the viruses in the system. That was done by Luc Gagnon at Nexalis. So we can see where this antibody ranks in comparison to the other 200 in the database. We can get its immune footprint, its profile. And this one was actually engineered to be missing those functions to eliminate any risk that it could enhance infection. We can see where it binds in that neighborhood group. And more work from Vamsi Rayaprolu and Teresa Govering in my lab show exactly what its structure looks like. Inside those yellow boxes, those are the microscope images of the arms of that antibody with its fingerprint-like recognition anchoring down onto the receptor binding domain of spike. We can also get temporal information. We can see that the three possible copies of the antibody don't seem to all bind at the same time. And when they do bind, they open up the spike. So this explains how we might get molecular synergy by these high resolution views. Now this was my weekend project. This is what I got up to on Saturday night. Can we predict, based on this wealth of data that we now have here in Buren Peters' database, how can we predict how well an antibody will neutralize SARS-CoV-2 based on all the different features that we have measured about it? And it turns out to some degree we now can. We can actually write an equation On the bottom, the neutralization is a beta knot plus a bunch of terms. And each one of those terms is the strength of different functions or features that we're measuring. So for example, we learn that you can predict whether or not that antibody is going to be effective in neutralization by where it binds. So this epitope group. So particularly if it binds at site 10 or site 3A, that antibody is going to be a winner. We can also predict how well it's going to neutralize by how tightly it binds, by whether it anchors onto that spike and doesn't let go. That's another key predictor. What does site 3A look like? That's CoVIC 10 that we just showed you. By anchoring onto that place and doing what it does, which is open up that spike, we can see how it is inactivating that spike, how it's blocking receptor binding, and how it might open up sites for complementary antibodies against other domains. And so that is the work that we are doing to understand at the molecular level how ideal immunity works against SARS-CoV-2. By building a global collaboration to bring together expertise from everywhere in the world and also everywhere in this building with the world's leaders that we have working here, we can build a broader, deeper, more durable data set to understand immune protection against SARS-CoV-2 and how to deliver it. So let's work on the molecular level. We understand those molecules, we understand those submicroscopic features, but how do you deliver that protection in a living thing? And that living thing is much more complex immune system. Well, to answer that question, I'd like to introduce my colleague, Professor Sujan Shresta. She and I share lab space. I'm at one end and she's at the other. Sujan's an expert in immune protection and vaccine protection and the complexity of those levels and how my molecules actually deliver the protection that we need in people. She got her bachelor's degree at Smith, her Ph.D. from UC Berkeley, did a postdoc at Washington University, and we're very fortunate to be able to recruit her here in 2005. She's a professor at LJI ever since. So I'd now like to introduce Dr. Shresta.
Thank you, Erica. Today, I'm going to talk about a variety of infectious diseases emerging and re-emerging that impact many countries worldwide. Until the beginning of this year, the focus of my lab has been on flaviviruses. These viruses include dengue, Zika, West Nile, yellow fever, and Japanese encephalitis. These viruses impact almost all subtropical and tropical regions of the world. We've been studying how these viruses cause disease and how the host immune system tries to control these viral infections using a variety of mouse models, cell culture models. And we validate our findings based on these model systems by looking at infections in people in endemic countries, such as Nepal, Thailand, Vietnam, and Japan. In the next 14 or 15 minutes, I'm going to tell you about our recent work towards development of vaccines against these viruses, dengue and Zika in particular. And then in the remaining minutes, I'm going to tell you about how we're using our expertise and knowledge from our flavivirus studies to contribute to the global, towards tackling this global coronavirus problem. So let's start with my favorite viruses, if you can have favorite viruses. These are these two viruses, dengue and Zika. They're very similar. They're transmitted by the same mosquitoes, these Aedes mosquitoes, and these mosquitoes are now all over California and right here in San Diego as well. These viruses cause a spectrum of illnesses ranging from mild to severe. The mild forms of these viral infections are called dengue fever and Zika fever. The severe form of dengue infection is called dengue shock syndrome, and yes, this can be fatal. Zika complications include the serious birth defects, which is now collectively known as congenital Zika syndrome. And I'm sure many of you remember back in 2016, pictures of babies with small head or microcephaly born to mothers who were infected with Zika while they were pregnant. In adults, Zika can also cause Guillain-Barré syndrome, which is this temporary onset of paralysis, and yes, this can be life-threatening. Towards understanding and develop safe and effective vaccines against these viruses, my lab for the past 10 years or so have been trying to address this central question, which is, how exactly does the adaptive immune response protect against these viral infections? Before I tell you the answer to this question, I'm going to give you a brief overview of the adaptive immune system. These viruses, dengue, Zika, and now CoV-2, when they infect people, they elicit two major arms of the adaptive immune system, B cells and T cells. You've actually already heard about the B cell arm because these are the cells that my colleague, Dr. Saphire, told you about that produce these antibodies that specifically recognize, bind, and neutralize these viruses. In contrast to antibodies, T cells, they recognize and directly kill these virally infected cells. So over the years, this is almost 10 years of research from my laboratory and others as well. What we've learned is that this adaptive immune response to dengue and Zika, it acts as a double-edged sword. In particular, this antibody response to this virus, they can actually play both a good and a bad role. Good meaning, yes, these antibodies will inactivate the virus, as we've just learned. But in the case of dengue and Zika, these antibodies can actually promote infection of cells because the virus now gains entry into these cells through these antibody receptors that are expressed in the cell. In contrast, work from my laboratory in particular have shown that these T cell response to this virus protect even when the antibody levels fall and they're no longer able to destroy the virus, or under certain conditions, these antibody response become bad and they promote increased infection of different cells. Despite this knowledge that the antibody response to these viruses under certain conditions can play a bad role, when Zika emerged in Latin America with its devastating consequences in 2016, 45 different vaccine candidates were developed, again, literally in less than a year. What's incredible is that every single one of these 45 candidates, they were focused in eliciting the antibody response, and they were targeting one viral protein. Here, E stands for envelope. Out of these 45 candidates, 8 of them have advanced to clinical trials, and they're currently being tested in people. In contrast to these 45 different vaccine candidates, our laboratory decided to take a different approach. We decided to target 2 different viral proteins, the envelope, the E protein, because this is, as I just told you, is the major target of the antibody response to these viruses. And we also decided to look at the non-structural 3, or NS3 protein. This is because this is one of the major targets of the T cell response to flaviviruses. We collaborated with Synthetic Genomics, a local company right here in San Diego. They used their Alphavirus-based mRNA replicon vaccine platform and created 2 different vaccines for us. One is expressing this NS3 protein of Zika, and the other one expressing the envelope, the E protein. And to make sure that this envelope was expressed properly, we also included a part of this precursor membrane protein. Now, in the next few slides, I'll tell you about how these vaccines behave in our animal models. The first type of experiments we performed was we vaccinated these animals, these mice, either with the PRME vaccine or the NS3 vaccines, and our control group were injected with saline. We gave them a booster shot, and 3 weeks later, we challenged these animals with Zika. 3 days after Zika infection, we looked for the amount of infectious virus in different tissues. The results are in the next slide. On the y-axis, we're looking at the amount of virus. And here, these are the 3 groups of vaccinated animals. Here, we're looking at the serum, and the brain this is one of the major targets of Zika virus. Each circle represents an individual mouse. we can see that in these control animals that were not vaccinated, that were injected just with saline, we see high levels of Zika virus. In contrast, mice vaccinated with one of these, either the PRME or the NS3 vaccine, we see either no virus or very low virus. We next wondered, are these vaccines good enough? Yes, they can limit infection, they reduce the viral burden. Are they good enough to protect against lethal disease? And here's the answer. Here in the y-axis, we're looking at the percentage of survival of these mice after Zika challenge. Control animals injected with the saline, 0% survived. That means none of them survived. In contrast, 100% of mice that were injected with either the NS3 or PRME vaccine, they survived. So what this result showed is that yes, all of these vaccinated animals can survive the lethal challenge. All these results that I just showed you, they were conducted following infection of these mice 3 weeks after the booster shot. We next wondered whether these vaccines were good enough to provide long-term protection. To answer that question, we vaccinated the animals as before. Instead of waiting just 3 weeks, we now doubled the length of time and we looked at 6 weeks of infection. So we challenged these animals at 6 weeks after the booster shot. In addition, we added one more group of animals. Here at the bottom, we're now vaccinating animals with both the NS3 and the PRME vaccines. 3 days after Zika challenge, we're looking at the amount of Zika virus in different tissues, and the results are again in the next slide. Here in the serum, brain and liver, the results are the same in all of these tissues. We're looking at high levels of infectious virus here in these control mice that are not vaccinated. So this is as expected. Now, in stark contrast, these mice that are vaccinated with both NS3 and PRME, we can't find any virus. In contrast, these mice that were vaccinated with just the NS3 or the PRME vaccine, we see that some of these animals now have these infectious Zika virus. What this result showed is this combination of two different vaccines provides a better long-term protection than each one of these vaccines alone. Based on this result, we believe that a flavivirus vaccine, such as that for Zika, should elicit not just the antibodies, but both antibodies and T cells so that we can maximize both efficacy and safety. As we were finishing those set of experiments early in 2020, the world changed. Towards tackling this growing coronavirus problem, we decided to forge collaborations with our already established partners in different countries. And instead of focusing on flaviviruses, we decided to turn our attention to understanding this dance between the virus and the human immune system by looking at coronavirus infection in different populations in these different countries. We're now part of the National Institute of Health funded research consortium called Centers for Infectious Disease in Emerging Infectious Disease, or CRID for short. Specifically, we're part of this CRID ESP. And the goal of this particular group is to investigate the epidemiology, surveillance, and pathogenesis of SARS-CoV-2 infections in all these countries here in Asia, Ethiopia, and of course, the US. Now, you're probably wondering why in the world are we conducting CoV-2 infection studies in humans in all these different countries when the US is the epicenter? The answer to that question is because this virus-host interaction, this dance between the virus and the host, is likely to differ depending on the population, the geographic locations, and climatic zones. We can ask the impact of climatic zones on coronavirus infection and transmission quite easily in a country like Nepal, where you can go from almost sea level to the Himalayas in a really short distance, 100 miles. And of course, Nepal is strategically located between India and China in South Asia, which has very high population density with a quarter of the world's population. And Nepal also shares similar cultures and, unfortunately, poorly functioning health system with its other countries, neighboring countries in South Asia. A key aspect of our collaboration in these different countries is that we are partnering and trying to establish mutually beneficial relationships with some of the best and the biggest universities in these countries. An example here is Tribhuvan University in Nepal. Here's my counterpart, Professor Krishna Manandar at TU, with his students here, and two of my postdoctoral fellows here at the front, who are conducting side-by-side research involving both flavivirus and coronavirus in collaboration with Professor Manandar and his students. And the reason for this is because we think it's really important to build local scientific capacity in these countries so that the scientists in these countries can tackle and contain these viral infections before they spread to other countries. And in doing so, not only do they contribute to their own country, but actually to the whole world by addressing these really important questions related to not just coronavirus and flaviviral infections, but Nepal is also a host of other important global diseases, infectious diseases of global relevance, including HIV, influenza, beyond infectious diseases, autoimmune diseases, cancer. So we think collaborations such as this offer tremendous opportunity for investigators in the US, right here at LJI, to train the next generation of global scientific leaders. Now that I told you about our studies going on with studying coronavirus infection in humans, you're not going to be surprised based on the data that I just showed you with our flavivirus work. We're also developing mouse models. And we're doing this through partnership with another local company, Symbol. And the goal of this partnership is to develop the most relevant mouse models of SARS-CoV-2 infection. These mouse models represent a critical tool towards achieving this CoVIC, coronavirus CoVIC goal that my colleague, Dr. Saphire, just told you about, which is to identify the best possible monoclonal antibody-based therapies against COVID. What is our role in this CoVIC consortium? We will use these mouse models expressing human antibody receptors, which are critical for controlling the half-lives of these human antibodies, so that we can test the safety and efficacy of these human monoclonal antibodies against COVID. And lastly, but not least, you won't be surprised that we're also working on vaccine development against these coronaviruses. We're super excited and we're thrilled that these 3 different vaccines have provided greater than 90 or 90% or greater efficacy, at least short term. And these are antibodies focusing mainly on, these are vaccines focusing mainly on eliciting antibodies, and they're targeting a single viral protein. So potential limitations based on our understanding of virus-host interaction from our flavivirus world is that we don't know at the moment whether these first-generation vaccines will provide long-term immunity, long-term protection. We don't know if they'll protect all individuals in the US and the world. And as I just told you, the antibodies under certain conditions, at least for flavivirus antibodies, they can play a bad role. We're also wondering, as this vaccine-induced antibody response wanes, do we have to deal with enhanced disease severity upon reinfection? And of course, as my colleague, Dr. Saphire, just mentioned, we're constantly aware of these viruses, these RNA viruses in particular, changing, mutating. So we wonder, will these first-generation vaccines continue to provide protection against different strains? of not just CoV-2, who knows, we may have another newly emerging coronavirus in the near future. To address this potential limitation, my laboratory has been developing coronavirus vaccines that elicit both antibodies and T-cell responses using multiple viral proteins and immunization strategies. And one of our goals is also to develop a pan-coronavirus vaccine that we hope will work against not just SARS-CoV-2, but also closely related coronavirus, including the common cold. In conclusion, I have taken you through, well, my colleague, Dr. Saphire, and I, we've taken you through viral structures of the coronavirus to antibody-based therapies. Then I brought the T-cell immunity. I talked about developing vaccines that elicits these two complementary arms of the immune system, both antibodies and T-cells. And I told you about how we're developing the next generation, the state-of-the-art mouse models for testing these vaccines and antibodies so that we can protect diverse populations, not just in the US, the whole world. How are we doing that? We are forming these multidisciplinary global collaborations that are mutually beneficial, both for LJI, my lab, and my counterparts' lab in these other countries. And we're doing this through these incredible consortiums here. We are applying, again, these complementary approaches of eliciting both arms of the immune system, both antibodies and T cells, and we're targeting multiple viral proteins. And by doing so, we're developing, LGI is at the forefront of developing, again, complementary tools or approaches to tackle the global COVID problem. We're involved in developing both vaccines and testing and identifying the best possible antibody-based therapies. Thank you for your attention.
Thanks, Sujan. Before we get into questions and answers, I just wanted to let everyone know that we're going to go a little bit past 1 o'clock so that we can get through some Q&A. Before I go there, if you could slides up, I would like to cover something it's very important and perhaps understated, but and that's the role of philanthropic support. Here at the Institute, we're a nonprofit 501c3 organization. And when the pandemic first began, the normal thing and the normal structure and frankly, all things normal were turned on their head. And what came and made the difference was philanthropic support. Many of you who are on the webinar today are supporters, and for that, I sincerely thank you. And I just want to remind everyone some of the differences that funding makes. The first is it's fast. Philanthropic support can answer the call immediately. And those of you who have provided gifts to the Institute, we're not side. You weren't passive observers to what was going on, but you actually got in there and helped us make a difference immediately. Second is the directness of those gifts. Philanthropic dollars can be deployed immediately. They can let us respond, which was absolutely critical. And if you look at the work that was done here at the Institute in the early days, and the speed at which that work was put into the public domain to educate everyone from industry to the public about what was going on, what to expect immunologically. Some of those papers came out of the Institute, and that work was funded philanthropic support. It's catalytic. We have had a leadership gift that have allowed us to upgrade infrastructure, to rapidly build out our biosafety level 3 lab, to implement new instrumentation, and to really make sure that we were capable of doing the work that was separately needed right away. Even to today, we're expanding our clinical core facility. This is where donors, in this case, blood and tissue donors, come into the Institute. we're able to collect those samples right here at the and deploy them into the research labs with delay. And finally, perhaps something that's not that often enough, and that is philanthropic support provides the fuel and encouragement to my colleagues here at the Institute who I've seen work tirelessly, day, night, weekends, only looking for a chance to do bold things, to make a difference, to really ahead of things while still mattered. And when philanthropic dollars come in, we feel that sense of participation, that sense of help. There's a certain pride in showing to our supporters that those gifts are well placed here at the Institute. It's a legacy. I hope that as you back, you say, I did something during the pandemic. It's made a difference, and I can point to it there at the La Jolla Institute. We still have needs. We're looking to expand our ability to do proteomics as an technique, and we've actually made inroads into recruiting a candidate, and are on the last pieces of a cane to be able to fund the purchase of an instrument that allow us to look in a very unbiased way across the entire proteome, all the proteins that are produced in investigating health and disease. And so with that, I'd like to switch gears and go into questions and answers. Some of them have been answered online, but I did want to get through a set of them with our panelists. And the first is a summary of many questions that have come in and in part answered, but I'd like each of the panelists to just quickly answer the burning question, and that is, are vaccines we've heard of safe? Should I wait to get them? So let's start with Mitch.
You want me? Are vaccines safe? Yes, I believe that they seem to be safe. They've been tested in tens of thousands of people. Obviously, they're going faster than in previous cases. The previous record for vaccine development was 4 years. But these are undergoing a rigorous process. What do we lack now is long-term follow-up. I would guess that for an RNA-based vaccine, the long-term follow-up, and here I'm speculating, isn't going to show any more adverse reactions than we've seen so far, and they are very, very few. The third contender, if you will, the AstraZeneca vaccine, is virus-based, adenovirus. And there have been very few adverse events, and not sure they're related to the vaccine. And adenoviruses have been used experimentally for a long time, and they're likely also fairly safe. So I feel confident about the vaccines, but we lack long-term follow-up.
Great.
Other comments?
Yeah, Erica?
I'm on some of these committees, and a lot of the acceleration is just purely administrative. Rather than getting in a package of data and then convening a meeting 2 weeks later, we convene that, we have 24 hours to read it, review it, and then we convene the meeting. We're also not waiting to fly to a meeting. We're doing it by Zoom. We're also trying to remove other barriers. Like let's just assume vaccines are going to be approved. We're going to need glass vials. We're going to need refrigerated trucks. We're going to need distribution systems. Let's get those all figured out ahead of time. So removing those administrative delays doesn't change the safety. The complete safety profile and detailed analysis still happen. We're looking at hundreds of pages of data.
Great. And Sujan? Sujan, you're muted.
Okay, I agree. These vaccine data have been examined thoroughly by world experts all throughout the globe. And right now, the clear unknowns are some of the ones that Mitch highlighted, and I pointed in my talk too. The durability of this vaccine-induced protection. Will it protect all different populations in all different countries? And even in the US, maybe certain populations will not be protected. And, but I'm super optimistic because we have these different vaccine platforms, not just RNA. And when I heard that the AstraZeneca vaccine also has high efficacy, that's really good news. from multiple perspectives. We want different kinds of vaccines for different people. Some people may not be able to take one or more vaccines. And in terms of the supply chain, we don't want the vaccine where they all need the same critical components to make these vaccines. So it's great that we have different vaccine platforms, different companies, all working towards getting these approvals.
Thank you. Yeah, I'll just finish up real quick with the practical answer, and that is waiting a vaccine is essentially sign yourself up to get COVID. The prevalence of the pandemic and its worldwide spread means if the vaccine is available and it's effective and it's safe, it's a hedge against what might happen otherwise with a wild infection. So I agree with panelists. So now I'm going to ask a couple more questions, and I'm going to go through the panelists here. I guess, Erica, we've kind of beaten this one to death, but there are questions about maybe sheltering in place to wait it out. Is there a chance that things would just fizzle out?
Good question. So if a virus is only human, and it only spreads human to human, and there's no animal, there's no insect, there's no other vector, then isolating all the humans will do that. This is why we can eradicate smallpox. It only has a human host. You vaccinate all the humans, you can erase the virus. Now, what about coronavirus? It spreads human to human, for sure. And if you could actually isolate everybody in a box or an apartment by themselves for the maximum incubation plus the safe period, let's say 3 or 4 weeks, and they stayed in that department, they had food for 4 weeks, nobody left, you could eradicate it. But in a practical term, that's kind of impossible. There are places in the world where people can't socially isolate, they'll starve to death. There are people that still need to work in hospitals. There are people that still need to run the power plants. There are people that need to deliver the food to your house. And so the fact that we are still out there to some degree, lessened, but there are people still out circulating. And the extent to which human behavior is and isn't careful and accelerates or reduces viral transmission, it's going to circulate. And the other important thing is that humans aren't the only things this virus infects. We think it spilled over from bats. It has now spilled from humans to mink. It is on multiple mink farms. It has spilled back from mink to humans. There are multiple other species. We always interact with animals, whether they're agricultural, livestock, pests, mice in our house, things that birds, insects, mosquitoes. The extent to which this is now endemic in the globe in multiple species, that horse is out of the barn, the cow's out of the barn, whatever the expression is. It's too late. You can't stop it anymore.
Thank you. So Mitch, both of the presentations talked about this a little bit, but because there were so many questions, can you kind of describe what preconditions or medical therapies might preclude people from getting a COVID vaccine?
Yes, maybe I can answer that in the context of exclusion criteria in the original trials. People getting immunosuppressive therapy, children, pregnant women, these are all individuals who may not be eligible for the vaccine. For example, for children, you'd want to test them separately, and that hasn't been done. Everyone knows that as a rule, children are less symptomatic, but there are some children who get a multi-inflammatory syndrome that can be very serious. So vaccinating children could be important, but we don't know if the vaccine is effective in that group. I would think any immune deficiencies in particular, or people on immunosuppressive treatment, which can include bone marrow transplants, certain treatments for autoimmune disease, probably wouldn't be vaccinated. And those individuals, aside from social isolating, maybe beyond when most of us would as the pandemic trails down, those are the kinds of individuals who'd be most needing and most eligible for the kind of antibody therapies that Erica talked about earlier.
Thanks, Mitch. Sujan, there have been some questions that have centered around, if we have vaccines for humans, aren't we done? What's the need for animal models and further investigation like that?
Great question. we need animal models because we need to be able to understand how vaccines work. Right now, we have only the first generation vaccines, and we're all bringing up the point that it may not last long, it may not protect certain individuals, certain populations. To make vaccines better, we need to understand how they work. And the way we do these vaccine studies and understand how they work is by testing them in animal models, because we can take out components of the vaccine in this immune response, add back different components of the vaccine immune response, and figure out what are the molecular and the cellular players of the immune system that's contributing to vaccine protection. And obviously, we can't do those experiments in humans. Thank you.
So it was answered in part in the QA online. So I'll take this one with some help. But there were several questions asking. We were collectively surprised by the effectiveness of vaccines, and answer that by I was pleasantly surprised at the results. But I think that as an immunologist, it's not so much surprise that we can respond to a vaccine. The real questions are, in the end, its efficacy in preventing disease, its long-term protection, and perhaps one of the most important question is, will it stem future epidemic proportion outbreaks? In other words, will vaccines provide the kind of protection where not only are you protected, but you're unable to transmit the virus? And I think all those studies really speak to why the work continue and why we're going to try and understand. Because certainly there will be sense of the population that won't get the vaccine, it won't work. And we think globally about it. Was I surprised by the effectiveness? I was pleasantly surprised. But I think that there are a lot of really important questions that'll really seal the deal as to what the vaccines will do from a public health standpoint. So switching gears back to Erica, you mentioned it a little bit, but do you think that mutant strains are a threat to our vaccine strategy in minks and COVID? So I think the answer is yes. But talk a little bit about how strain mutations can matter both with COVID and otherwise.
So viruses are error prone when they replicate, and they are constantly changing. Now, most mutations are detrimental. Those are great. We like those. Some confer an advantage to that virus. And so as it samples one host or a different host, whether it's a human or a mink or something else, it's adapting itself to succeed in different cells. And so it picks up changes. Single changes could knock out single therapies. A single change will probably knock out a complete immune response. So an immune response to your vaccine will be what we call polyclonal. You'll have those millions of antibodies. You might have lost a couple in that million, but you have all the others. So if there are just a few changes, the vaccine will still work. What we would worry about are individual single antibodies that hit one place. If that mutates, we've lost it. That's why we want a two-component cocktail, and that's why we want ones that hit places that can't change. We also want to evaluate the number of changes. And this is why we need a new flu shot every year. There's a lot of flu in the world, and as it spreads through millions of people, as coronavirus is, that's a lot of rolls of the dice. And that's a lot of mutations that pick up. And so over time, it drifts. The flu does something else the coronavirus doesn't do. Flu's genetic material is in multiple different pieces, and they can shuffle like cards in a deck. And so flu can have bigger jumps in its change. That's why we need a new flu vaccine every year. It's going to be some time before we need a new coronavirus vaccine, but I do think that we need to be on top of what those mutations are, because they're changing how that virus behaves. And I've got 5 of my scientists hammer and tongs on that very issue right now, so stay tuned. We'll have those results for you.
Thank you. So Mitch, I'm giving some zingers today, and here's another one. Recent results, and us unable to be sure. Why do you think a lower first dose might be better than a high first dose, as has been reported so far by the AstraZeneca vaccine?
Why don't you ask me a hard question? So this was described as a head scratcher. Um, First of all, I want to say that the lower dose was a mistake. It was done in about 2,800 or so individuals, approximately. And then they increased it, and a much larger number got the full dose. If it holds up and turns out to be real, we have to think about that. It's possible the lower dose more closely mimics natural infection in terms of how you normally get infected and make an immune response. One thing we might say is that developing immune memory, especially for B cells in the germinal center, takes rounds of cell division and differentiation and takes some time. And if you get a very high dose, it's possible that you don't get the chance to do that, that the existing set of the trillions of antibodies we all carry around, even though they're not really optimal, that with a very large dose, there's enough of these kind of weaker antibodies that they can control the virus, and you don't get a chance to develop immune memory. Frankly, that's a hand-waving explanation. But one more thing I can add, there are some adenovirus studies where a lower dose or waiting a longer time between doses seem to improve the immune response. So I hope I didn't injure myself with my hand waving. Thanks.
No, I think it's beginning to be about that time, but I wanted first. Thanks for that. It's tough commercial data, especially when it's not reported in scientific literature. But I'm going to end with a question for the panelists, and it's a combination, and that is a two-parter. The first is, can you get different vaccines? Maybe there's the first one now, and then another one later. Are you going to be brand loyal, for lack of a better term? And then as the second part answer, the question: should you get vaccinated if you've already had COVID-19? So I'm going to start with Erica, and then Sujan, Mitch, and I'll back clean up.
I'll start with the first question.
Okay.
There won't be enough immediately for everybody, right? So the good question is, if it's not available, you're not going to get it. If it is available, you need to be high priority. You need to be elderly, immunocompromised, high risk, or frontline medical worker. You may not be able to have choices. You may not be able to go to the CVS and say, well, I don't want the Moderna, I want the Pfizer. You might just have the one that's available. There might be certain geographic regions that get one versus another. It's going to be some time for production to ramp up. And remember that each vaccine has 2 doses, one now and a booster a month later. So when they say they're making 50 million doses, that treats 25 million people. So we might not have as many choices as we are used to in the marketplace, but they're all very similar. They're all RNA encoding the spike protein or a vector delivery of the spike protein. And so they pretty much will work the same way.
Jim.
My inclination is a little different from Erica's. That's because we do these vaccine studies, at least in animals, and one of the parameters of vaccine-induced immune response and protection is we combine different vaccine platforms and see how the immune response changes. So in the absence of data where these companies have not done this, what we call this heterologous prime boost types of experiments, I would stick to the same in getting vaccines from the same company. But that's definitely a really important approach that I'm sure many labs worldwide are trying to see if combination of, say, the Moderna vaccines followed by the AstraZeneca vaccine will give you a better, a little longer-term protection. So I think it's quite exciting, these results by different companies and different vaccine platforms.
Well, I'll take on the question of should you be vaccinated if you've already have had COVID-19, assuming you're convalescent and you're better. So you'd certainly be low priority. The problem is that we don't know how long protective immunity lasts. We do know that people who've had mild cases do have, at least at the 6-month level, and some of that has worked from our institute and as well as others, that some of those people have signs of long-lasting, meaning 6-month immunity. So, and it doesn't have to be a severe case. So my tendency of my thinking would be that if you've had the disease and you're convalescent, meaning you're well, that you're a very low priority for a vaccine. And as we collect more information about how long protective immunity lasts, maybe you would be getting a booster after 5 years. Think of the tetanus shot. You get a tetanus shot every 10 years or so to keep you protected. So it might be on that order, but we just don't have enough information yet to go out beyond, say, 6 months because this is such a new infection. Definitely, you'd be low priority.
Thanks, Mitch. So I guess I'll just I'll mention that if I found one sort of truism across everyone is we like being locked up and hidden away, and I think vaccines are well, we don't have to. We know that vaccines are essentially the most effective public health measures ever created. And if we were transported back to the early 1950s on the verge of polio vaccine, I doubt that there'd be a conversation about really want one of those. And while the scale and difference that we have in our healthcare system gives us hope that there's more there to help us, I would like to just have a parting thought that next Thanksgiving will be here, next Christmas. And part of that formula will be the immunity that is gained in our population, and vaccines are going to be a huge component of that. So I'll get vaccinated, and I'll look for data to see how long it's going to work and how well. And if there's another one, switch teams after Sujan lets me know which two work best together. And science is going to tell us the answer. We're going to know these things, and it'll take some time. But certainly, given the opportunity, getting vaccinated or just seeing what the device gives you with COVID, the disease, doesn't seem to make sense. So with that, we're over time, and so I don't want to take anybody's time any further. This presentation, the Q&A, it'll all be available online, and if we could roll the end credits, there will be slide here that'll have contact information for getting in touch with the Institute for donations or otherwise. And a sincere thanks to everyone who attended, and wish you all well. Happy Thanksgiving, and keep your masks on when you're out.